Tonight's speaker is Cal Soul. He's with the Field Museum. Cal is a postdoctoral student who works closely with Arjan Maan. He is going to give us an update on what the latest information on CT scans and electron microscopy, with focus on the Mazon Creek concretions, if I understand that everything. I met Cal at a recent fossil field trip, and I am really looking forward to tonight's presentation. And everybody seems to know him, so I'm not going to keep blathering on. I'm just going to turn it right over to Cal.
All right, well, thank you so much for the invitation. I'm really happy to be speaking here, and I'll just start sharing my presentation. And of course, I want to thank everybody else as well for inviting me here and giving me this opportunity to speak to everyone and share a little bit of what I know. Um, yeah, first off, my name is Cal. I did my undergrad, well, I grew up in Madison. I went to college at UW-Madison where I majored in zoology, geosciences, and conservation biology. Last year, I actually graduated, well, I defended my dissertation last year at George Washington University, where my advisor uh, was Jim Clark. And so now I've started doing postdoctoral research with Dr. Arjun Mann at the Field Museum. And I met a lot of really cool people along the way here, including at the University of Wisconsin Geology Museum, Dave Lovelace. Highly recommend visiting it if you get a chance. I think I've spoken to some that have been there before. So, yeah, share the share the awesomeness of the UWGM and go local. But anyways, so as a researcher, what I'm most interested in is the origin of the modern day. What is the context of it? There's hundreds and millions of years of vertebrate evolutionary history, and as just one person, I can't possibly know it all. So I choose one, and among all the modern vertebrate groups, I'm most interested in studying modern amphibians to try and answer that sort of question of how do we get to today? And apply a three-pronged approach to my research where I study anatomy, and using that anatomy informs our understing the evolutionary relationships, and together they come together and inform the change in ecology and shift through an evolutionary pattern through time for these different lineages. So, the origins of amphibians. Who are the amphibians? Today, we see them represented as frogs, salamanders, and the elusive Sicilian. And these are all very diverse groups, and they are represented by a huge diversity of body forms, and they occupy a diverse, a breadth of ecological niches. So yeah, they not only do they live in like ponds and lakes, streams and rivers, but they also live, some can live underground like the Sicilians for, yeah, most of their lives. And some amphibians also spend their entire lives at the canopy of trees. And so to sum it up, they are incredibly diverse, and we expect to find a lot of them in one place in the modern day because of how much how different they are, how they can occupy different niches because of their different specializations. And personally, I'm a huge fan of amphibians, and I keep 8 newts as pets currently. I used to have 14. And you can see a little video of one of my tiny little newts coming up to the front of the tank here and acting all pretty. Anyways, more about lissamphibians. Lissamphibians, or otherwise known as the living modern amphibian group, they belong to an ancient lineage, and molecular data dates them back to originating at least 330 million years ago. which if you scaled all of evolutionary Earth's history into a clock, um, we would we would find them right around there, to 330 million years ago in the late Paleozoic. At this point, it's the Carboniferous. And another relatively interesting way to think about it is that the time between the earliest amphibian relative and the first dinosaur, which is 100 million years, is way longer than the time between the last Triceratops and present day. So this is an incredibly ancient lineage and has so much potential to unveil our understanding of all this time leading up to modern day. And so what's also crazy that the earliest lissamphibian fossils, the earliest frog, salamander, and Sicilian fossils already have near modern morphology and anatomy, and they show up as early as 251 million years ago. So that's about 80 million years of missing evolutionary history between the earliest lissamphibian fossil and the estimated divergence time of their origin in the Carboniferous. And also because of this, because of their the sparsity of the paucity of their fossils and how they're small, they're really difficult to preserve. They're biased in the fossil record, biased against the fossil record. With all this lack of fossil data, we have even less understanding of how the unique styles of modern amphibian development evolved, like the metamorphosis, the classic, like you have the tadpole or the newt larvae, and then it changes, grows limbs, loses gills, so on and so forth, loses a tail in the case of frogs. And so hopefully with this introduction, you have some takeaways here where this is a very ancient lineage, and there's relatively little change between like today and the earliest lissamphibian fossils. Some might even call it, I don't know why they're not, but they probably should be considered, living fossils, as controversial as that term is, considering how little they've changed and how old that lineage is. They're super diverse today. We can find many different species in a relatively small area. Think about your local pond, local body of water, local stream, and there's just a lot of amphibians, frogs and salamanders there at least. Once again, as an ancient lineage, they have this potential to contextualize how a lot of land vertebrates, including us, change through time, or at least our ancestors changed through time in the Carboniferous. And so as ancient lineage, we're not really sure about how some of these changes in their anatomy led to their modern morphology. We're also not really sure about the changes in their development through time that led to the current, metamorphosis strategy of modern day amphibians. And we're also not sure of like what were the ecological changes that led to their known diversity. Like how did they get to this point where there's so many amphibians you can find in one area, they occupy like all parts of the rainforest, all parts of them, like the redwoods. They're nearly everywhere, found on nearly every continent except for Antarctica. So what were the changes that led them to this current amazing diversity? So fortunately, we have a lead to figure out and fill in the gap in our understanding of their evolution. And we have some ideas of some fossil tetrapods that might help contextualize the origins of modern day amphibians. So who exactly are these evolutionary predecessors that are the forerunners to modern day amphibians? There are actually many candidates. In other words, there are many hypotheses for the origin of modern day amphibians, and we're going to go through these hypotheses. So first of all, we have the what I like to say the amphibamiform hypothesis. Simplest to say is that modern day amphibians, they all stem from a group known as the amphibamiformes. There's another hypothesis that suggests that modern-day frogs and salamanders stem from amphibamiforms, but modern-day caecilians, these long worm-like amphibians, they stem from a different group known as the stereospondylus. And finally, there is this other hypothesis that suggests that modern amphibians stem from another group known as Recumberostra. And really, nobody cares about Recumberostra. This hypothesis, I'd say, is debunked essentially. And so we're going to focus on these two hypotheses, the amphibamiform hypothesis and the other one, which is, I don't know, people have a lot of names for it. But the one thing in common that they have between these two hypotheses is regardless of whether they come from one group or two groups, these groups all fall within the temnospondyls. And so temnospondyls are amazing. Has anybody here heard of temnospondyls? Just show of hands, or say something in chat. Yeah, it's a relatively uncommon thing to know about in the paleontology world, and that's what's so great about temnospondyls. Like, I can say anything I want about temnospondyls, and because I'm the expert and you invited me here, you have to believe me. But anyways, like modern-day amphibians, they have a diverse array of body plans. They came in many shapes and sizes, and they lived all over the world. But they also we have fossils of them on Antarctica. And also to think about how these animals are related to modern-day amphibians. Temnospondyls are to modern-day amphibians as dinosaurs are to birds, this larger prehistoric group that the modern-day group is part of. And so you might think, like, just like how dinosaurs inform our understanding of birds, temnospondyls can inform our understanding of modern-day amphibians. So now that we have the hypotheses, where do we even begin if we want to cover the who, what, when, where, and why, and how of modern-day amphibian origin? Now, I'm going to tell you a little bit about my journey exploring these questions. So in my undergrad, I was really interested in the caecilians. So once again, these are these long, worm-like, legless amphibians. They're completely different from what you imagine, typically of frogs and salamanders, right? And they're so elusive and mysterious even today because they're burrowing all the time. They live underground, and it's like difficult for modern-day biologists, wildlife biologists, to keep track of them and study their behavior because so much of it is happening where we can't even see. But they're also really interesting in terms of their evolutionary history because we don't have a lot of fossils clarifying how they got their, long snake-like body, snake or worm-like body, and how they lost their limbs as a descendant as a member of the tetrapods. And so in 2017, there was a huge debate about the origins, and one of the hypotheses that I mentioned earlier was at the forefront of this debate. Did caecilians come from this other group separate from modern-day caecilians or modern-day frogs and salamanders? As an undergrad with the University of Geology Museum, I went a lot to Wyoming for fieldwork every summer. It was great. And I had the experience of discovering a new species, as part of the story. So in this part of Wyoming, near Du Bois, which is like west central Wyoming, you can walk along this ridge and you can find some of these interesting circular structures, round structures in the exposed surface. And all around this ridge, in situ and also eroded, excavating some of these structures, you can find these round, these cylindrical shapes deriving from those round structures. These are burrow casts. And as any good paleontologist would do, if you think something's a burrow, if you think something's inside, you crack it open. And inside these, we actually found, skulls of some sort of animal. And it took a couple and we prepared a couple of fossils and eventually described it. These were animals known as Ninibihan ducutica. And this is a fossil that I worked on describing since I was an undergrad and recently published on last year. There's an interesting story concerning its name where we worked with the Eastern Shoshone tribe of the Wind River Reservation, did a lot of outreach with them and a lot of partnership to share knowledge with their Fort Washakie school. And out of that partnership, the name for this taxon was one of the products. We're still having that ongoing partnership, and it's been great to develop paleontology and develop, exploration of fossils in the Eastern Shoshone community. And so here we have it, Mnimia dugutica. I described it. That basically means I did all the taxonomy and the anatomy of it. So I identified it as new species, described how it's looked and the paper. And the next step, like I started looking at what it might be similar to, we found that it's related to, using phylogenetic analysis, we found it's related to these other fossil amphibians known as fossil stereos found in the Chinle and Riley Millers. And that, fulfills how we're trying to use these fossils to understand the relationship of this species with other known species. And we were really excited. Like this group of animals, like in 2017, were like, these are related to caecilians. That's what, people were debating over. Like we had an opportunity here to be like, we found a missing link. And once again, as part of the three-pronged approach to research history, also looked into their ecology and their evolution. And so because these animals lived in a time when Wyoming was really hot and dry for most of the year, like even hotter and drier than today. And they risked a lot of desiccation. They were basically in this arid desert, and they would only receive like the monsoon rains once a year. So how did these animals survive desiccation when, like, for the majority, 90% of the year, it was a desert? Amphibians, they need water, otherwise they'll dry up. And so we found a lot of evidence that these burrows were their estivation burrows, where they essentially dug a hole, and they resided in a state of torpor, kind of like hibernation, and wait for the next rains to show up and wake them up, and they can start swimming around again. So yeah, that's the ecology and evolutionary story of this fossil taxon. So in the end, did we end up finding the missing link for the ancestor to modern day caecilians? No, unfortunately. It was fun while we were studying it, but long story short, there was very little support, and other fossil discoveries weakened the support for this hypothesis. And so that is basically my introduction to investigating the origins of lissamphibians. And you're probably wondering why. What does any of this have to do with Mazon Creek? And so here's when we get into the last hypothesis that we haven't talked about. So it's most likely that all modern day amphibians descend from these group of fossil amphibians known as the amphibamiforms. And this, for the past decades, has honestly been receiving the most attention and has been the most supported. And so the amphibamiforms are a really key group if we want to understand how modern day amphibians evolved. And so that leads me to the title of our talk today, the taxonomic diversity and development of late Carboniferous amphibamiforms from the Mazon Creek Lagerstätte. And it is here where you find the oldest amphibamiforms. And so I'm really lucky to be able to study at a place like this, where so many people, the community, such as ESCONI and other avocational paleontologists have contributed to this sort of research. Unfortunately, because of some, as scientific research is always updating or understanding new things, I won't really be able to get into the development of these the amphibamiforms of the Mazon Creek because I found out new things, and I need to work on those observations in order to make the best interpretations for as a product of this research. But anyways, the Mazon Creek Lagerstätte is really famous for the earliest amphibamiform, Amphibamus grandiceps. This was described by Edward Cope back in 1865, and on the screen here we have all that's left of the holotype. And I say all that's left, just this, illustration, because the holotype has since been destroyed in fire, and we don't have it anymore, unfortunately. But there's always a silver lining. We have found other specimens of what we believe to be Amphibamus grandiceps, and so we've named a neotype for it, and we've since really been using the neotype as kind of the foundation for identifying an animal as Amphibamus grandiceps. And we've done a lot of work with the neotype. A member of our lab, of the Man Lab, Miranda, has been working hard to reconstruct the fossil in 3D. And she'll also be reconstructing other Mazon Creek amphibamiforms in 3D. So please stay tuned for that. And so a lot of concretions containing these amphibians have been discussed, described, and referred to as Amphibamus grandiceps. And some were actually initially considered different species, but they've since been synonymized with Amphibamus grandiceps. So now we have this idea, like all of these different concretions that have these amphibians in them, these, we're going to say they're Amphibamus grandiceps. But there was a lot of conversations between paleontologists that haven't been published, like doubting that these were all of these fossils, all these concretions represented only one amphibian species. Because unlike modern day settings, you expect to find multiple amphibian species in one community, right? So why would we find only one Amphibamus grandiceps, one temnospondyl, or one amphibamiform in Mazon Creek? And knowing like how diverse modern amphibians are, is the modern diversity restricted to the modern day amphibians? And the older stuff, the amphibamiforms like Amphibamus grandiceps, are they not very specialized? Do they only occupy like one singular niche? And only later on did that lineage evolve the diversity that modern day amphibians have? So that's a very huge question that we're trying to investigate by looking at all of these fossils. But thinking about modern day amphibians again, we have these two salamanders. These are from Northern California, and they can be found living quite close to each other. And they're among many, many more species of salamanders in the area. And so, looking at them now, would you say that these two salamanders are the same species? Yeah. Catherine says no. No. And Andrew says no. Yeah, Bruce. Yep, everybody's saying no. Everybody agrees, right? So then would you say these two amphibamiform fossils are the same species? No, yeah, nose all around. Yeah, in fact, it's quite different. So it is, in fact, a new species. It is not Amphibamus grandiceps. This is a new species that we're currently describing. It's been donated to the Field Museum by Dave Douglas. And in our biology analyses, we've actually found this is the closest amphibamiform relative to the rest of modern day amphibians. So it is a very key fossil in understanding that turning point between amphibamiforms and the rest of modern day amphibians. We have other analyses that show this body plan is more salamander-like and less amphibamus-like, which means, around 309 million years ago, a salamander body plan has already evolved. And so, did I skip one? No. And so, studying the taxonomy, anatomy, the systematics and phylogenetics of this this fossil, we identified a new species and found that, like, in terms of their evolutionary relationships, is very much a key fossil in understanding amphibian origins. And so, thinking about modern day amphibians and their diversity in their body plan, it has this new taxon has a body that is very reminiscent of these slender salamanders that live in the canopies of the redwoods in California. And so if they share the same body plan, it's possible that they also occupy the same sort of ecological niche, and that maybe it's an amphibamiform that was very arboreal and lived in the tree canopies as well, whereas stuff like Amphibamus was living, more amphibiously in the waters or like occasionally treading onto land, but more like a typical salamander, like a tiger salamander that you might imagine. And so rather than considering that the current diversity of niches is exclusive to modern representatives, perhaps these niches have actually been occupied as early as the Carboniferous. And that's crazy, which means that everything that, not everything, but what if all of these niches that modern day amphibians occupy currently were something that was protractedly obtained over their long evolutionary history? And that's something what my research right now on the Mazon Creek concretions is seeking to understand. And so, despite being the oldest amphibamiform so far, they are already very specialized for different niches. Some that, we have the long-bodied taxon that may be living in the tree canopies, and then we have, the Amphibamus grandiceps that may be living more terrestrially. And so there's a little bit of a story of the ecology and evolution. And we've developed like many more evolutionary hypotheses, ecological hypotheses that we can further explore. And so the things to take away from this is that, maybe there are there may be more new species, which hint, yes, there are, lying in wait, hidden and requiring extra taxonomic attention to better detail and understanding and to better detail and understand their identity. And so, and also perhaps we're underestimating the ability of amphibamiforms in the Carboniferous to explore and occupy a multitude of niches that may have been the origin of the niches for modern day amphibians. And so there are also other amphibamiforms living in other parts of the world. So something that my colleagues, including Arjun and I, have found in previous analyses is that we have a similar story where there are all of these amphibians coeval living in the same place at the same time in the Permian of Texas. And these also, based on our analyses of their skull shape, have the potential to occupy different niches. And so perhaps there is some relationship between the ability to occupy different niches and the origin of modern day amphibians. And there continues to be other areas of amphibamiform that we need to explore, such as these fossils of Eoscopus. I mean, these, under a closer look, you can already see that there is different morphology across some of these skulls. There's, skulls that have larger orbits, and then skulls that seem to have more horns, and different shapes and sizes. And so maybe after all of this, I kind of sound like this, where I'm like, there's got to be something there. But what I really mean is there's a lot of work to do. And yeah, excited to do all that work. Thank you all for listening. I have a lot of people to acknowledge, and the list goes on.
So where in Mazon Creek were these amphibian concretions found?
I believe they were found in various parts of Pit 11. Yeah, I can't remember the locality that off the top of my head.
The whole
So
thing
there's a
comes
chance.
from the river.
Oh, the river, really? The original holotype? The
Yeah, the
river?
Joseph Evan was the one who found it, I believe.
Oh.
And he's the one that had it when it burned up.
Oh.
Very interesting. I'm it now makes me want to go back and look through, not that I'm going to really see anything, but through my other concretions and say, well, I thought that was this. Maybe it's not. I know, wishful thinking, wishful thinking.
Well,
Stop
there's
laughing at me, Alan.
Yeah.
no, that's the thing about, the work we do in paleontology, is it's not just wishful thinking, but with more observation, with more information, that may
Right.
yield better detailed information that may shift like how we perceive some of these fossils. And that's kind of the story
Finding
that's been going on here.
Oh, hey,
finding
Cal,
something in the future that shows you what you had in the past and can go back and realize what you had originally seen.
Exactly.
hey, Cal, of all the millions of concretions that have been processed, do you have an estimate of how many have been amphibamus or animals like amphibamus, I should say, have been found? How many
I I don't think I can estimate that. I mean, that must be, I can imagine that proportion is very small relative to all the concretions that have been cracked open, and that's only considering the Mazon Creek concretions. In France, there's a whole nother similar operation that happened where people were coal mining and there's a lot of spoil piles, and there are also a lot of concretions. I think you and I have talked about this a lot, Rich. A
Yep.
lot of concretions have been produced as a part of that excavation, and those also have amphibamiforms. So that's also another thing that could be taken into account when estimating, how many amphibamiforms have we found.
Hey,
So
Cal.
what are the size of, like, the one I didn't pay attention,
Oh,
but the real small one? Uh,
um, let's
yeah,
see.
back, you were just there. Go back. There on the right.
On the right. Oh, I
Yeah.
don't have a good scale on that. I forgot to put a scale on that one because that was just an iPhone picture. But it's relatively small. I think the skull is shorter than a centimeter long.
Yeah, I think the specimen's about 4 centimeters long.
Yeah, because it looks small, and I'm like, wow, I could see somebody definitely misidentifying that
Yep.
as
As
something
a
else.
fish. Yeah.
Yeah, because I keep thinking about that one that you thought is a fish. Maybe it's not a fish. Maybe it is one of
Mhm.
these.
Yeah, and this specimen from Dave Douglas's collection, it is also one of the things that made me reconsider, the development of amphibamiforms and look into that a bit more. Um, that has been the wrench in that part of the narrative.
Hey, who knows? Maybe some of my coprolites are actually these guys.
Hmm.
Oh, well, I
Yeah.
know that Arjun and Jason were looking at the larval tetrapods, which I think were pretty misunderstood or even ignored or missed altogether before
Right.
recently. So once that paper becomes sort of public, I think people have to re-examine their collections.
Yeah, yeah. Yeah, I'm not sure, what had been historically discussed, but it does seem like a lot of things ended up being lumped together, and this may have been a part of this philosophical differences that paleontologists had over time, where uh, there's kind of like waves of like people thinking, paleontologists thinking like, oh, it's these things are, we are, too zealous with describing species, like think about all the modern diversity that we have today and how many of the members of the same species just look different, that phenotypic diversity. And then, there's another wave of like, wait, let's take a closer look again. Let's actually like do the taxonomy and find like, anatomical evidence to support it besides, just like general description. And so
Hey,
that's what we're finding here.
Cal, are there technologies today? I mean, obviously micro CT scanning, SEM, et cetera, technologies that are helping you separate species from one another?
Honestly, it's been good old counting the vertebrae. Yeah. Bruce, I think you had a question.
Yeah, I did. It just struck me, that these seem to have very much a body form that has gone through to modern times or very closely. Yet here we are at 306 million years ago. They didn't just appear here. So what's prior to this?
Exactly. That continues to be the question.
A Tully monster.
Mhm.
Because like I said, as this whole assemblage is very diverse. Like we have the idea of like amphibamus, and we use amphibamus to compare to others and find that like the long body taxon is like looks different, and we think relative to amphibamus is very specialized. But who's to say amphibamus is the general form
Right.
either? It itself might be really specialized for terrestrial locomotion. Um, and and then there's stuff like brachiosaurs that have specialized in a different direction to be more aquatic too. So what preceded all that? That's we, the narrative continues to shift further and further back.
Right, right. And it just, we haven't found a location where these are preserved.
Do we have any of these from the Triassic record?
The Triassic record, in terms of amphibamiforms,
Right.
we
Yeah.
do have, yeah.
That would fall into that camp.
There is only one amphibamiform taxon surviving until the Triassic, if you exclude lissamphibians. And that's Micropolis doi from South Africa and Australia. No, not Australia, Antarctica.
Oh, okay.
So-
What was the one that was Chinley something or other?
Oh yeah, Chinley stegophus is part of this the group known as stereospondylus, and so that is as its name implies, is a Triassic taxon.
Yeah, that's what I thought.
Yeah.
Okay.
So it's part of this. They're not amphibamiforms. They're part of a different group of temnospondyls known as the stereospondylus.
Right.
So I have a question. It's going to sound rather odd. Of course, a lot of my questions sound rather odd. But if some of these were terrestrial, has anybody ever found any of them along with the fossilized fern or bark or anything like that that would support that theory?
I guess like the best way to support its the potential of terrestriality is because contextual evidence can vary a lot. Right? Things can actually end up together, and if you look at it at face value, it might be a different story than what's the actual context. The word that we use for this concept is allochthonous or autochthonous, and autochthonous means that these things are coming from the same place, and allochthonous means that things have come together from different places. But I think the best way to test for their terrestriality or other occupation of other niches is to really know their anatomy and know how this anatomy and be able to compare it to anatomy of other things with, you know, potentially similar styles of locomotion or similar habitats. And some people have taken an extra step and started to apply biomechanical studies and other types of analysis to really test rather than confirm whether or not these things were terrestrial. Yeah, let me know if that makes sense.
Yeah, it does. Thank you.
Are any of these temnospondyls, are they known from Bear Gulch in the Mississippian? Or no?
Irrigal temnospondyl. No, I don't think so. I think when you get to the, let me double check. Let me Google this really quick. I don't even know.
Well, that's all right.
Everything I've seen from Bear Gulch has been
Fish and stuff,
largely
right?
marine.
Yeah. No, so in the Mississippian, we were really looking at stuff like Dendrocotton and Balaenoptron from the Joggins Formation or West Kirkton.
So they would have been around. They're just not known
Yeah.
then, right?
Yeah. And these things were already pretty diverse themselves. Like the story continues, you know, 330 million years ago, when Dendrocotton and Balaenoptron were around, they're already like.
Oh, so it even goes back farther. Those are just the earliest ones known.
Exactly.
Gotcha.
So when I was researching something, I remember reading where they thought frogs originated down in South America and slowly moved, you know, spread. But does that fit with some of the early forms that, you know, like the one you've discovered? Well, I guess I'm not sure what how my question is coming out in this one.
So are you asking, I mean, let me know if I'm interpreting this wrong, but you're asking, does the locality of the fossil record support this hypothesis that they originated from South America?
Yeah.
Yeah, that's a really good question. So this deals with the study of biogeography, which is a really tricky thing to do. There's a lot of nuance to it because biogeography is conditioned, like this analysis, most analyses, what we do in paleontology that produces the sort of result like ancestral reconstruction, which is, you know, you're estimating using statistics to find like the most likely ancestral state of something. These are all conditioned on the phylogeny or the evolutionary tree that you estimate. And so if you imagine if you include more taxa that live or are found in South America, that constitutes more of that statistical analysis. So that will be biased towards South America. And if you take away or you add more stuff from like Africa, then you shift that analysis to be more biased towards Africa. And so when you look at these analyses, you want to look at the sort of taxa that they're sampling and compare with and think about well, did they include anything? Did they exclude anything? So what I understand about frog evolutionary history is that the earliest frog from 251 million years ago is from Madagascar. And then you have stuff that's coming out of Chinle, you know, Triassic, later Triassic. You have stuff from Poland, which is also Triassic, but early Triassic. So these early frogs are pretty cosmopolitan. They're all over the place. So I would have to take a look at that analysis to let you know what I think about it. An interesting thing is if you're only looking at modern day frogs, the earliest diverging frog shows up is from New Zealand, which is very confusing if you think about it, because New Zealand is, as it is now, is relatively young. It's about like 50 million years old, I think, when, as it formed as an island now. So, it doesn't really make sense for, if you're just looking at modern day frog and you're only looking at, the pattern of the tree, to consider New Zealand as the origin of modern day frogs.
Wow.
Hmm.
Yeah, that would have been one of the last places I would have ever thought.
Yeah, and to be clear, it's not likely that it's, New Zealand, since New Zealand is relatively young. And so it needs to be taken in the context of as much information as we have to be more holistic.
Collection
Fascinating.
Collection what?
bias.
Right.
Yeah. Mhm.
So without additional samples, how much further can you go? What's the limit of our scientific ability to figure this out?
Yeah, that's a good question. And this ends up leaning more philosophical or epistemic, as like, using our data, how is it that we know what we know? Or how do we, how to use our data to inform our understanding, and what does that mean? And so a lot of people, methodologically try to apply new forms of analysis using different types of statistics. More complex methods sometimes include, machine learning algorithms nowadays. And, I'm not saying anything, whether or not it's right or it's wrong. But it is definitely something to consider, is like, when we're doing our analysis, what exactly are we analyzing? Are we analyzing the actual evolutionary history, or are we analyzing the data that we have now? So for me personally, I think I'm more reserved in my use of these more advanced methods, because I prefer to ground what we know, based on the evidence that's at hand. But these methods are still really useful in making hypotheses. So that based on what we know now, it looks like the origin of frogs might be in South America. But then if you find new fossils, that may be updated. So these things, I think, should be taken with a grain of salt. And but it is, if you interpret them as a hypothesis and acknowledging all these caveats, then these can be very powerful tools.
Cal, how
Any
many?
other?
Oh, I'm sorry. Cal, how many people are working on these ancient amphibians? How big
Yeah, so
group is that?
temnospondyls, I think there's definitely at least a dozen,
There's you and Arjan and
Oh yeah, a couple of Europeans, a couple of South Americans. A lot of people who did study temnospondyls have, uh, a lot of people who are foundational paleontology, like Romer, Bob Carroll. Unfortunately, they passed on and left, their students. But it seems like a lot of the students of their students have moved on to other groups. And so, at least in North America. In South America, I think there's a lot of interest in temnospondyls, and in Germany, there's a lot of interest in temnospondyls. But definitely far less popular than dinosaurs.
I'm gonna have to practice saying that word, temnospondyls. Yeah.
I wish there was a
need
better
to.
thing to call them.
That's kind of catchy.
Because it's not gonna roll off my tongue very easily. So I'm gonna have to practice it.
Yeah, it
It's a
doesn't.
cool word. It's a very cool word.
Yeah, it means temno means cut, spondyle refers to the vertebrae, and so the name is referring to how their vertebrae is composed of several different parts.
Hey, Cal, on a
Hmm?
completely side note, when I was an undergraduate at UC Berkeley, I had a terrarium with California slender salamanders and red-legged frogs.
There you go. I mean, you got amphibian diversity right there.
Yeah. So this and they were all from the canyon, above the campus.
Oh yeah,
So
that's awesome. That's really
yeah,
cool.
yeah, I kept them for a little while, fattened them up, and then returned them.
But yeah, no longer slender
No longer
salamander.
slender salamanders.
I have a South American horned frog right over there.
Oh my gosh.
Oh, those things.
He's
Yeah,
huge,
Big Mac
massive,
sized.
and I have to be very careful when because he thinks my finger is food. He's grumpy. I like him.
What
Yeah,
does
we've
he
got a
what
little-
does he eat?
He eats dubii roaches, which are really easy to breed. He will also eat little pinky mice. Yeah. So I'm one of those horrible people that
Buys live things to feed them to him.
Yeah,
that
we've
he's
got
cool. He's very cool.
we've got a little cabin in Arkansas, and it's been a couple years ago, but there's an area behind the cabin there that about half the size of a football field, and in that one small area there, I could find like 10, they may not all be different species, but at least 10 different color patterns on salamanders.
Oh my.
Wow. The
And so
frog loved cicadas last year. Ooh, he thought cicadas were just awesome.
Didn't have to go to the pet store.
I
Yeah.
don't go to the pet store anymore. I've got enough. Yeah.
Well, that's after you harass the guy about dead fish and things, right?
Yeah.
Ah.
Thank you so much, Cal.
Yeah, happy to be here, and yeah, thank you for
Very
inviting
good.
me.
Yeah.
Thank
I
you.
think a lot of people are going to be looking back at their concretions and looking more closely to see if
Yeah.
some of their things might not be what they thought they were.
Mhm. Yeah, I'm happy. I'd be happy to hear what people find out. And it sounds like we're signing off, but before everybody signs off,
Oh, we're not. We're probably
okay.
going to just stop the recording,
Oh, for
and
sure.
then we'll let the conversation. We can go on as long as everybody wants to go on, but we'll stop the recording. So
Will do.
Well, we had to laugh. We had one of our people came in and he had was going through our collection and he said, "Oh, he goes, I used to own this. I used to own that."